Hello, and welcome to our presentation on NTRES, a biological sequence database query system. Let's get started. So I'm going to start by speaking about biological data in general. And the central dogma of molecular biology is that the code of life, DNA, gets transcribed to RNA and then to protein. Proteins are what do work in your body, and so they're highly studied molecules. Each of these are sequences, meaning they're made up of building blocks from an alphabet, and the order of the sequence matters. And frequently, these sequences will inform biology researchers on how to design experiments. So researchers will read previously written peer-reviewed journals along with their associated data, which could be sequence data and more other data, and design their experiments based on that. And from those experiments, they will generate a new peer-reviewed article, which also has new discovered sequence data and other data. And so all of this generated data needs a place to be stored, which used to be very difficult before the advent of computers. But in 1965, Margaret Dayhoff created the first publicly available database of protein sequences called the Atlas of Protein Sequence and Structure. Since then, many databases for sequences and other associated biology data have been created. And a way is needed to consolidate all of these into one search system, because if you're writing a new research paper, you want to make sure that, first off, the research hasn't already been done, and secondly, you're finding the best hits for whatever it is you're looking for, and you didn't overlook something that may exist or has been previously published. Enter NTRES. NTRES is a metadatabase, which is a database that stores metadata, so data about data, from all these different biology databases in order to have one system to query all of them.
RefSec acts as an interface for a wider range of biological databases. This allows researchers to query multiple databases simultaneously without needing separate tools for each. Additionally, NTRES links related records across databases. For example, a gene entry in RefSec can be connected to its sequence in GenBank, related literature in PubMed, and associated variations in DBSNP. The databases and tables under NTRES are all linked, and therefore the results from one query can be pipelined into another to increase efficiency and reduce duplicate queries from the database. Key tools from NTRES include eSearch, which is used to search for a gene and returns an ID, similar to selecting only the ID column that matches a condition. eLink is used to link between different databases or records, similarly to a join. eFetch is used to retrieve the data for a gene, like a select, except it returns data in the format of an XML, JSON, FASTA, summary, or another data format. Since NTRES handles large amounts of data in queries, it must have scalability features to maintain growth. It uses indexing to organize data, as well as pre-computing relationships between the tables and databases, allowing for fast retrieval even when querying massive datasets. Additionally, queries can be batched, processed in parallel, and have the quantity of results be constrained for faster response times. Moreover, the data returned by NTRES' API can be compressed, such as in a gzip format, to minimize bandwidth usage and speed up data transfer, as well as have the commonly requested query results be cached on servers, which allow for queries to be answered without needing to use a database. As shown earlier, it is possible to query with high specificity by using the gene name, which is the primary key, for a fast computation and a smaller result download, as well as keep a local cache of results to query locally using extract, which removes the repeated queries. There are many databases under NTRES, but the largest and most commonly used one is the Huge Genome Assembly version 19, NCBI RefSec. Therefore, that is the database that will be investigated. The HG19 NCBI RefSec table serves as the central repository, and there are fields in it which are shared amongst many other tables. Bin is an indexing field for range queries, which is used internally to speed up searches for genomic ranges. It organizes genomic regions hierarchically for efficient lookup. The name is the primary key, it's the name of the gene, and it acts as a key between most of the tables. Chrom stores the chromosome or scaffold name, which is the reference sequence on which the transcript is located. Strand is either positive or negative, which is used to express whether the strand orientation of the transcript is positive or negative. TX start stands for the start of the transcript, or for if the strand is positive, or end if the strand is negative, using a zero-based index. TX end is the opposite. It stands for the end of transcript for a positive strand, or the start of the transcript for a negative strand, and it uses a one-based index. CDS start stands for the position of the region where the coding sequence begins. If non-coding, then it will match the TX end. Similarly, CDS end stands for the position of the region where the coding sequence ends, and if it's non-coding, it will also match TX end. Exon count stands for the number of exons in the transcript. Exon start is a comma-separated list of exon start positions using a zero-based index, and exon end is the comma-separated list of exon end positions using a one-based index. Score stands for the score of the transcript, which was used historically but has lost usage in modern times. Name2 stands for alternate, more human-readable name for the genes. CDS start stat stands for the status of the CDS start annotation, whether it's none, unknown, incomplete, or complete. Same for CDS end stat, it's the status for the end annotation. Finally, there is exon frames, which stands for the reading frame of the start for the CDS region of the exon. It is the direction of the transcript 0, 1, or 2, or -1 if there is no CDS region. As mentioned earlier, there are many tables in the HG19 database. The all RefSec curated and RefSec select tables all contain genome information, the curated one containing manual reviewed curated genes, and the RefSec select containing high confidence, but they all have the same schema. There are additional tables, such as the NCBI RefSec CDS, which contains the coding sequences associated with RefSec entries. There's the HGMD table, which links the RefSec data to the Human Genome Mutation Database. There's the link table, which links between RefSec records and external databases. There's the PSL table, which stores the alignment data for RefSec sequences in PSL format. And finally, there's the sec NCBI RefSec table, which contains a session for the sequences in RefSec.
There are a number of refinements which are used by the NTRES system which allow for queries to happen much faster and to yield better results. Some of these refinements are like those we have studied in class, with biological databases using a number of indices which make the general retrieval of data much quicker. Some of these refinements are more unique, like heuristic querying, which is specifically tailored to yield fast results when looking for matches in DNA sequences between large data sets. NTRES optimizes biological data searches by using indices based on standard attributes like protein names, properties, and unique accession identifiers. These indices allow for quick data retrieval within a database. To unify queries across databases, NTRES creates pre-computed similarity links. These links function as unclustered indices, allowing connections between otherwise isolated datasets. This system ensures that even with varying database structures, NTRES provides fast and uniform search results across the databases. When querying biological databases for matches between N sequences, traditional dynamic programming methods construct an n-dimensional alignment matrix to find the best sequence match. While this approach is thorough, it's time-intensive and grows exponentially slower with sequence length, with a time complexity of O(m) for sequence length m to the n for number of sequences n. Heuristic querying, on the other hand, approximates results by performing a quick initial search to identify regions which seem to match, which are then explored in more detail. Though it sacrifices a bit of precision, heuristic querying is significantly faster. To show the significance and the difference between the time taken by dynamic and heuristic querying approaches, a script is created which generates two sequences of simulated biological data up to length 50,000 and then searches for the best match between the two using both a dynamic and a heuristic approach. At a length of 50,000, the dynamic approach takes just over 11 minutes, while the heuristic one takes less than the 10th of a second. In one of the research papers which first proposed the heuristic approach for this use case, a biological data query went from taking 12 days under the dynamic implementation to just 12 seconds under the heuristic one, with comparable accuracy in the query results. BLAST, one querying tool for Entrez databases, refines heuristic searching through several methods. Seed matching focuses on short, exact matches to serve as starting points for further exploration. The two-hit strategy increases accuracy by requiring two seed matches close together before advancing and searching further in the region. Finally, substitution matrices use evolutionary knowledge to prioritize meaningful matches, discarding those which don't seem to hold any evolutionary suggestion of a true match. These refinements make BLAST a powerful tool for querying extensive biological databases with speed and precision, and further refine the optimizations used within Entrez databases for biological querying.
Alright, so I'm going to walk through a quick feature demonstration of the Entrez CLI, showing off some of its core utilities and their ability to be directly piped into BLAST in order to efficiently and quickly query biological databases. So here's the command that I'm going to be walking us through, how it works. It's just split into some lines with the escape character here to escape the new line. So the first e-utility is eSearch. What this command does is it searches the specified database with the -db. So we're searching the PubMed database here, and our query is for lycopene cyclase. And then we're going to be piping the output of this command into the next one. So our next command is elink. This is one of the core utilities of Entrez, and it's really what makes it unique. What this one does is it uses a library of pre-computed neighbors to take the output of the previous command and return an enhanced set of query results based on those neighbors. So we're using the -related field here to indicate that we are linking to related papers in the PubMed database that have been pre-computed. So next we're going to be using elink again, but this time we are targeting protein databases. So what this will return is a huge list of protein sequences that are related to the articles were returned by the previous commands. So next we're going to be using, oh, there's a little typo there. That should be eFilter. My bad. We're going to be using the eFilter utility, which filters down the results of the previous command. We're specifying that the organism we're looking for is a mouse, and we're specifying the source of the protein sequence to the refsec database. Next, we're using the efetch command, which downloads the query results in the specified format. So we're choosing FASTA, which is pretty much the standard for protein sequences. Next, this is actually just a Unix utility that takes only the first two lines of the returned output. I did this because I didn't want the commands to be running for like 15 minutes. So what this does is we're only going to be using the first result of the previous command. So only the first sequence in FASTA that we've downloaded. So now we're able to pipe the resulting FASTA protein sequence directly into BLAST, which is very useful. So we found the protein sequence that we'd like. Obviously, this is not very realistic. We wouldn't just randomly pick the first one that was downloaded, but this is just an example. So we're going to be searching using BLAST. This is the -query- is just making sure that We're using the output of the previous command as the query, and we're searching in the NR database. So remember, this database is actually, it's about 595 million protein sequences, which is quite a few, to say the least. The dash remote just specifies that we're querying the database that is stored on the remote database, on the remote servers at NCBI, instead of a local one. So you can actually download the databases yourself, but I didn't want to download the however many hundreds of gigabytes NR is. Then we're putting the results into the results.txt file and filtering for results that have an e-value of 0.001 rather, for the similarity. And the output format is 6, which is just it's the tabular format. So the output of this command is actually just a table of sequences that BLAST has deemed to be similar to the one that we're querying. And it only took 3 minutes to find the list of them, which I think is pretty good considering the size of the database we're querying. And I think it really highlights the effectiveness of the heuristic approach that BLAST takes, as well as just the overall effectiveness of the database architecture. So this has just been a quick example of a use case, like just to show off the workflow that you can use with this DBMS. So thank you.
In summary, computational tools have been indispensable for organizing biological data. Querying biological data against databases of biological data is in itself informative in the form of sequence alignments, whether they're global or local. And the algorithms in which we use to do sequence alignments in these queries is very important for the computational cost of querying these databases. Using the NTRES system allows a cohesive, consolidated way to query many different biological information databases. So hopefully these systems can continue to improve and we can learn more about life itself and develop better medicines, and our computational power will grow and the world will become better for it. Thank you.